Hello, fellow cultivators of curiosity. I'm Natalie Kaiser, your guide through the captivating universe, where seeds, genes, and everything in between converge. Whether you're in the field, orchestrating a symphony of polleny in the nursery, or decoding the secrets in the genetics lab, you've just stepped into the epicenter of scientific discovery. Jhon is a PhD candidate at Michigan State University, combining phenomics and genomics to predict grain yield and Fusarium head blight resistance while understanding the genetic basis behind these traits. When he's not deep into research, he's active in scientific and nonprofit organizations, and he's also a huge horror movie fan. So, Jhon, do you want to kick us off and tell us the story about how you navigated to your position now and really highlight, those options that you had along the career path and what made you make the decisions you did to land where you are today?
Yeah, well, first of all, thank you for the opportunity to be here. NAPB always has a special place in my heart, so anything for NAPB. So for me, ending up in plant breeding in general is a series of self-realizations, if you will. So it started when I was in high school. This was grade 8. So we had biology, and of course, biology encompasses a lot of things. But when we ended up in the genetics part of it, we were doing tri-hybrid cross by hand. So that was really interesting for me. And I'm like, okay, this is cool. It's not heavy in math, but it's also, very sciencey. So that really grabbed my attention. Although initially I wanted to be a medical doctor. So I that's why I pursued biology as my undergrad. But of course, me loving genetics is always in the back of my head, or it's always there. However, when I was taking my biology courses, I realized that I am really not that good in animal physiology and animal anatomy. So I'm like, I will be a really bad medical doctor if I pursue this. But what happened was I really loved plant science when I was taking my biology course. And for some reason, I felt like, oh, I was really good at it. And when taking my genetic subjects in undergrad, that's when I really fell in love of the notion of working on plant science and genetics at the same time. Luckily, I was able to get an undergraduate thesis in a research institution in the Philippines. So it was on genetic diversity of rice mutant lines. So I was doing phenotypic and genotypic diversity, which is kind of like my opening salvo in the world of plant breeding and genetics, basically. And then after that, I was hired in that lab, in that institution as a researcher. And from then on, I just got involved in more plant breeding work, breeding lines, selection, population development, field trials, marker-assisted selection, and I fell in love ever since, and I'm not going to any field anymore. So that's kind of like the gist of how I fell in love and how I ended up in plant breeding. Now, I ended up in Michigan State for my PhD uh, because I actually fell in love with what I will be doing. Because when we're looking at PhD opportunities, sometimes, of course, we email professors that might align with our research interest. However, when my current PI was telling what he's going to do on phenomics, on genomic prediction. I've never heard of phenomics before this, actually. And for someone who really loves taking photos and he's telling me that images will be the main source of your data, I'm like, let's go, I'll do this. So that's basically how I ended up in my current position here at Michigan State.
I think you touched on a point that's really hard for a lot of folks going into grad school is how to choose which program will be the right program for you.
Mhm.
Were you entertaining other projects or other programs at the same time? And can you speak a little bit more to how you made that choice? Was it really just the draw of getting a deeper dive into phenomics that pulled you to Michigan State, or how did that decision come about?
Yeah, you're right. I was actually also like looking at other programs, and quite honestly, I was also accepted to two other programs. But what really draws me to this is me wanting to learn something new. Because, like I said, prior to my PhD, I've never heard of what phenomics is. I've never done genomic prediction, and I've never done GWAS at that point. So that was a huge driving force for me. Wanting to learn. And of course, I knew going into PhD that I need to have something that I know I can use in the industry more. Or I mean, when I go to actual plant breeding. So, and that's actually what I wanted to do is on the more applied side of things, more on the line selection, more on the field trial as well. And the fact that I could use these tools and these approaches to do those things really draw me into that position basically. Yeah. But it was a hard choice.
Yeah.
I'm gonna tell you that.
So maybe for somebody who's not familiar with phenomics, could you give us the layout of what your day-to-day routine looks like? What kind of tasks and what kind of skills and capabilities do you need to have?
Yeah, in phenomics per se, of course, disclaimer, I'm not yet an expert on this. I'm still learning. Um, but the main part of this is one, being familiar with the sensor, because there is a lot of sensors and actually platforms. So in terms of sensor, you have sensors that could get RGB images. So that's taking data from your red, green, and blue light, similar to how our usual cameras work in our smartphones, in our digital cameras, in our DSLR cameras. Basically, that's RGB imaging. Now, on multispectral, you add two more wavelengths in there, or sometimes three. So primarily that's red edge and near infrared. And for hyperspectral, you now get from visible light to near infrared to shortwave infrared, basically almost the entire wavelength spectrum. And for infrared, that's primarily on the long infrared range. And what you're going to get is usually uh, thermal signature. So first is being familiar what type of data each of these sensors is going to give you, because that will dictate you, is it going to fit with my objective? Is it going to fit with the crop system that I have or with the plant species that I have? Is it going to fit with the trait that I wanted to explore? The other one is the platform that you're going to use. Are you going to use UAV? Are you going to use it in a field scale? So that's why you would want to use UAV, or is your trait a little overhead, like your plant height, your canopy architecture? But let's say your trait is more on the plant level itself, so you can probably use a stationary one, a handheld one. In the laboratory setting, of course, it's a different setup and system as well. So knowing what type of platform to use is also very important. And I think the most important thing after those two is image processing. And quite honestly, this is where I hit a huge wall when I was starting with phenomics. Because there's a different there are multitudes of approach on how you process the image. But the most important thing, regardless if you're using QGIS plus R, or if you're using Python, or if you're using MATLAB, or if you're using ArcGIS, the most important thing is for you to separate, let's say, your plot, or your plant, or your seeds, basically the feature of your interest, and getting the data out of it. So those are some of the crucial things in there. And down the line, of course, that's where your uh, knowledge in statistics will come in, your knowledge in prediction modeling will come in, and your knowledge in machine learning could come in as well if you want to go that route. So I think those are just some of the key things we need when we're talking of phenomics.
All the things you mentioned, the sensors, the platforms, the analytic tools are constantly evolving.
Yeah.
How do you ensure that you stay relevant and current and on the cutting edge?
Yeah. I think the most important thing with that is always checking the recent publications that are related to your field, which in this case, phenomics. Quite honestly, Google Alert is a good thing for this. Like, Google Scholar always being updated to certain keywords. And actually, that really helped me a lot. Like, every now and then, I'll receive emails of, publications related to my work. And, that's how I get uh, informed of what new studies are out there. The other one is always looking at phenomic discussions, both in Twitter, in Discord, Stack Overflow, stuff like that. That will really, because usually we exchange, you know, insights, we exchange knowledge. Oh yeah, this company has this new sensor. They tried it this way. Maybe this could also fit to your objectives. So those are the things. The other is every now and then these companies will give seminars, will give webinars. I always subscribe to them so that if there's something new that they're developing, or if they wanted to spread out the word about these new sensor, I will also get informed about this. But the most important thing is actually being personally involved in wanting to learn about the advancements, not just in phenomics, but in any field in general.
which can be a pretty big time commitment. So,
That
like
is
you
true.
said,
That is
you have
true.
to be dedicated.
Yeah, which is why I would stick with my suggestion of having your Google Scholar alerts and updates in your email. So when you check in the morning, you'll see that there's something new in the field. Yeah.
So, speaking of something new in the field, you just recently published a paper in Scientific Reports using phenomics and imaging technologies to better understand the genetic landscape of Dawn accumulation in wheat.
Yeah.
Can you tell us more about the raw data that you generated and then how you took that raw data and transformed it into meaningful phenotypes that you could associate with Dawn content? That's often the most challenging aspect, is to translate the raw data into a trait.
Yeah, that is 100% true. So I'm just gonna backtrack a little bit on how that project uh, came about, basically. What happened was we have the hyperspectral. I'm laughing already with this story. What happened was we have the hyperspectral camera for another project, which did not see the light of day. And the camera is just there sitting for a day or two, even three. And we rented that camera for, I think, $2,000 or $3,000 for 1 month. So I'm like, I'm wasting some resources here. I have to do something about this. Luckily, one day, one of my lab mates was processing the images, our grain samples that will be sent out for dawn measurement, because we usually routinely measure deoxynivalenol for to do our selections for FHB resistance, and we send it to a different lab to do GCMS. So when I look at the samples that she was processing, it's clear that there is discoloration in there. The seeds are different, have different shapes. There's chalkiness. There's some red pigmentations in there, and it kind of clicked you right away that okay, the differences in here could be measured by this hyperspectral camera. I'm just gonna worry about the processing down the line. But I need to take the images while I have the samples and while I have the camera. Did a quick search if people are doing this. Apparently there are, especially there was a group in Germany that did that, although this was a single kernel perspective. But anyway, that's a different story. But that laid down the foundation with what I'm doing. So went to my supervisor right away, pitched the idea to him, and right then and there he said, Jhon, the lines that you're imaging actually have genomic data. So you have to find a way now how you can combine your phenomic information that you're going to get from the hyperspectral data with the genomic data already present. So my mind went two ways. One is for GWAS, and the other one is for genomic prediction. So let's talk about the genome-wide association one. So the phenomic data is composed of 204 wavebands. And I think the number of genotypes that I used in there is around 300. So that's a 300 by 204 matrix of data. Now, essentially, each of these wavebands could be considered as one phenotype. But of course, I cannot do 200 for GWASs plus one for the actual DON content. That would be really time consuming. And actually, that would also be redundant since majority of the waveband, especially those that are nearby, will have really high correlation and there is a huge collinearity existing in that phenomic data. So the first approach that I did is to separate the first 20 wavebands, do principal component analysis to dimensionally reduce that information, and then use that first principal component as my new phenotype. And then I moved my binning or my separation by 5 wavebands, so there's an interval of 15, and do that until I reach my 1000 nanometer. So I ended up having 34, if my number is correct, 34 PCs, which of course is a huge reduction compared to 204. Although there are studies that used feature selection to identify which single wavebands they could use. But I stick with my principal component or dimensional reduction approach since I'm thinking maybe there are some information that could be lost if I go that route. So that's the first data handling that we did for the phenomic data. And then we were able to identify some marker trait associations, some SNPs, if you will, that are associated to specific waveband ranges, which is interesting. So that means specific waveband ranges may have different genetic control. But what's more interesting is that across the 34 binned wavebands, or basically across the entire phenome, there is one MTA that's always appearing, and that's the one in the long arm of 2D. So to verify that, what we did is, okay, let's dimensionally reduce everything. So I did PCA for all the 204 wavebands, again, used the first principal component as my phenotypic trait, and to my surprise and to my delight, it's the only MTA that was mapped using that. And when we looked at the DON content of the genotypes bearing the homozygous A allele and the homozygous T allele, there is a significant difference. And actually, it's glaring between their DON content. So it was really cool to somehow indirectly identify that MTA or that gene, if you will, because that MTA is actually residing inside an F-box protein domain coding gene. So that's really, really interesting. And of course, it's one thing to have that result coming from a GWAS, but we wanted to validate that hopefully using some gene expression study, using some functional genomic study to really verify if what we found using the hyperspectral data or using the phenomic data is indeed controlling lower deoxynivalenol content in wheat. And if that happens, I'm going to be really happy.
Well, it seems like it's rare that it's such a nice, tidy story where you have an idea and you're able to execute on it and find something that's promising. So that's really encouraging.
Yeah, I still really find it both funny and lucky and really actually inspiring to a point. And that study that basically sprung out of nowhere became the backbone of my entire dissertation. So I'm really happy that happened.
That you seize the opportunity with.
Yeah, exactly.
Is there a correlation anecdotally with the colors that you can see with the naked eye and the waveband that ended up being associated with your trait?
I think there is. I think there is. Because primarily majority of the wavebands that have higher correlation with the actual DON content is in the along the reddish region. So we're thinking now that primarily the red light is bouncing off more in the DON infected kernels. So yeah.
And when you were doing your simplification and dimensionality reduction analysis, how many rounds of iteration did it take you to land on the sliding window approach? Did you first try a feature contribution approach? Did you start with this approach? Did you mutate over time? I'm always curious about the analytical process.
Yeah, I did try first the feature selection approach, like you mentioned. I did try that, but I only found a couple, and since majority of the feature selected wavebands are, like I said, in the red region, and if you draw or plot the spectral profile of all the genotypes, you see that there's still variation in the other regions that may not be captured by the feature selection approach. And quite honestly, it was another professor who suggested the sliding window approach. Because she has an understanding of some computer science and computational biology approaches as well. So she pitched that like, hey John, why don't you try to segment the different waveband ranges and see which MTAs could be found in those regions. So it's a mix of course, listening to more experts, or sorry, experts in the field, and also bouncing off of what I am working on currently or what I have experienced working on my data.
A little bit of test and learn and a little
Yeah,
bit
exactly.
of consulting.
Yes, yes.
And you mentioned there was a genomic selection component that spun out of this project as well?
Yeah, yes. So for that, of course, since like I said, what I want is to be using information in a more applied sense, then at Um, I look at that as an opportunity to test if I can use hyperspectral imaging data as well for prediction. So I'm treating each of those wavebands now as individual quantitative variables that I can plug in a prediction model. Now to have a apples to apples comparison, I use your usual genomic prediction models like those from BGLR, your random forest, XGBoost, RRBlob, and then just be straight up change the SNP matrix with my waveband matrix.
Mhm.
So in general, we see a higher prediction accuracy for dawn using the phenomic data. But there's actually another thing that stemmed out of that. Yeah, so another approach that we did to basically combine our phenomic data and genomic data is to do model ensembling. So I took the predicted values using the phenomic data and the predicted values using the genomic data and did just simple average. In some cases, we see a better genetic correlation and phenotypic correlation between the ensembled predictions with the actual DON content rather than just the ensemble of phenomics or just the ensemble of genomics, which is really interesting. And another approach that I did was to straight up use the predicted values from the phenomic data and using the genomic data, again, the predicted DON contents from those, and put them in unsupervised k-means clustering and let the model separate my genotypes if they have high or low DON content. And to my surprise, it was actually able to separate those. So I think those approaches are really cool if either you want to predict the rankings of the DOM content of your selection candidates using both phenomic and genomic data, or straight up use that to narrow down your selection pool right away. So those results are just really cool. And to your point earlier, I do like when, things just fell into place.
Yeah. You mentioned that you really like to have something that has an applied purpose or, an application. Do you have a story from either your PhD or from your earlier career where you really had an impact in the plant breeding industry or world and you felt like you had real world results?
Yeah, so in my first few years as a junior breeder, if you will, in the Philippines. So I was very fortunate to be part of a breeding team working on abiotic stress tolerance in rice. And like I said, I was doing some marker assisted selection, some phenotyping in there, and some field trials. One thing I do like about that is some of the lines that I helped evaluated became varieties. So now it's being commercially available in the Philippines. Some farmers are now using it, and I think that's where it stemmed why I really want to be in the applied side of plant breeding, why I really want to be in the breeding population development, in the line development pipeline, or in the field trial pipeline, because I really want to see that product along the way up until it reaches the farmer's field. Um, with my work right now, I'm hoping that the prediction approaches that we're developing or we're pitching could help breeders alike do their selections quicker or faster or even more accurate. So in a way, I could be indirectly, helping product development at some point. So yeah.
What were the differentiating traits with those lines, the rice lines, and what were the breeding targets in that breeding
Mhm.
program?
Yeah, so for that, we were doing drought resistance, submergence tolerance, and salt tolerance. Uh, Specifically, I was doing submergence tolerance and salt tolerance. Although for salt tolerance, I'm just doing marker assisted selection. My teammate was doing the phenotyping for that. Uh, and our goal is to develop not just a rice variety that could withstand one abiotic stress, but across all three. And those varieties that were developed and released as a variety either has primarily drought tolerance with a little bit of salt tolerance and submergence tolerance in there, or you have primarily salt tolerance with some level of drought tolerance and or submergence tolerance in there. So basically our goal is to develop multi-stress tolerant rice varieties that of course have substantial yield under those stress conditions because at the end of the day, farmers would still want to have their yield so that they will profit.
Were you using an index selection approach, or were you pyramiding and phenotyping in parallel for all data?
Yeah. So for drought tolerance, we are using some index approach. One of my teammate was doing some principal component analysis in there here and there to have her selection on a multitude of drought tolerant traits. In my case, with submergence and salt tolerance, mine is on the marker assisted selection approach. And we are also using scoring approach to assess the phenotypic uh, performance of these lines under submergence stress and salt stress.
Did that
Yeah.
involve doing a lot of wading through fields?
Yeah, yeah. That's actually one thing I do love working in the field as well. Because in my case, I do field performance under submergence stress. So I flood my trial. After 1 week, I remove the water. And it's really nice to see some plots recovering, some plots are not. On a line selection perspective, you can clearly see the strips of tolerant lines and not tolerant one. And that's just, I think, is really interesting. It's really cool, at least from my perspective.
Me too. That's my favorite thing when you have that
Yeah,
visual selection and you get to see the progress that you've made.
that is true. Yeah, that's really satisfying.
So as a student yourself, do you have any input on one thing that you would change in the way that plant breeding students are educated today?
Oh, only one thing?
All right, you
No,
can
I'm
do
just
three. You can
I'm just
do three.
kidding. Uh, on a technical side of things, since right now we're living in an era of data, and regardless where we are in the breeding pipeline, if you're in the pre-breeding, if you're in the breeding line development, if you're in the trial or multi-environment trial, commercial trial part of it, we're always handling huge amount of data. So I feel like a little bit of data science training could go a long way for plant breeders. Of course, not to the extent that we're going to be actual data scientists, but for us to just be comfortable in handling data, in looking at data and thinking, how should we approach these types of information? And of course, that ties up with our statistical backgrounds and knowledge anyway. The other thing is maybe a little bit of coding lessons here and there, and also it ties up with my data science comment, because we so far I think we cannot just do away without a little bit of R coding and now maybe Python. But of course, we don't have to be experts in coding. We have different tools anyway. Like you have your Stack Overflow to look for things. You have even ChatGPT to help you build the codes. But what I'm getting at is for us to have that sense of uh, familiarity with the coding language a little bit. So that's on the technical side of things that some courses I would want plant breeding grad students to have. On a more professional skill side of things, I feel like one of the things that plant breeding graduate students, or actually graduate students in general, should have is science communication. And that's from designing your poster to presenting your poster, making your PowerPoints appealing not just to technical people, but also to people outside your field and maybe even outside of STEM. And also how you should be communicating to different audiences. I've learned that from experience, from working with high school students as their mentor, from working with farmers, talking with them during field days, to presenting at various conferences. And what I figured is that in each of these different audiences, I'm always explaining basically the same thing. But I need to be able to really shift gears depending on my audience. And for graduate students, you cannot always have that breadth or length of experience to train that. So I feel like maybe just a semester on scientific communication or include that in our professional development courses, that will be great. Because I feel like some of the miscommunication or misinformation happening in science stems from a little bit of lack of scientific communication training from the scientists. That's a sad reality, but I think that we will all benefit if we have that kind of training.
I wanted to go back to the technical piece that you
Mhm.
mentioned because I think so much of it stems from messaging. I think that
Yeah.
when you speak to a grad student about data science, if that's not
Mhm.
in their wheelhouse, it can be
Yeah.
intimidating. But I
Oh
think
yeah.
you framed it as just equipping you to be a good thought partner so that maybe if you're in a meeting or in a collaboration with a data scientist, you can ask the right questions to help co-create the project. I think that's an important lens to view it from. We don't need everybody
Yeah.
to be a data scientist, but we
Exactly.
need to be conversant.
Yep, exactly. That's the point that I'm trying to say. And also that stems from what do you call this? From experience as well when I started working with these huge amount of data. Like I have to have just a little bit of understanding or just enough understanding about how I should work with my data and actually contact my data science colleague like, hey, I want to try this approach. Do you think this approach will be good? And sometimes I even consult her with, this is what my accuracy looks like. Are my codes actually correct? So I'll send her the code and she will tell me, okay, this part is wrong. You can do this, do that. So I think you're right. Like just having that confidence and having that enough knowledge for you to really steer the conversation and really steer that um, collaboration would be really good for us. Yeah,
But it can be a vulnerable spot to be in. Anytime
that
you
is
have to
true.
share your code, you're really put on display.
Yeah, but But quite honestly, I was just talking to another professor a few days ago, and I told him, I'm not a good coder myself. I may be working in prediction modeling, but I recognize that I need some help doing this. So I think that's really an important thing. Like you said, we don't have to be data scientists right then and there to be able to do these things. We just have to have the knowledge of it, or like I said, enough knowledge of it, and know how to collaborate with the actual experts in these field,
Yeah,
and also use the tools that we have right now.
yeah, I completely agree. Um, yeah, maybe on the science communication piece, what besides pursuing opportunities that you mentioned, different extension opportunities, how do you hone your skills if they aren't incorporated currently in a formal curriculum?
I see. A big part of it is one, presenting in conferences, because there you're going to be exposed to, of course, it may be uh, people who are also in your field, but they're all on a different level of understanding. So there are probably some undergrads in there, some graduate students who are either starting or far along in their program, and of course, some professors. So that would actually help you train how you communicate your science really well. And quite honestly, right now, I think it's becoming a thing, these rapid talks. I think at some point as a grad student, you have to try it so that you'll be trained how to properly convey your message straight to the point, because you only have 3 minutes, kind of like a elevator pitch, if you will, of your research. I think that's a good and important training. And but the most important thing with attending these conferences is that you'll gain more confidence in talking to other people, regardless if you're talking about your science, regardless if you're just there to do networking. Anyhow, you need to build that kind of confidence. And quite honestly, this is probably something unorthodox, but always talk to your Uber drivers or your Lyft drivers every now and then. Especially, but when they start asking me like, what do you do? I cannot always go, yeah, I do genetics, I do hyperspectral imaging, genomic prediction, because even for me, it might go over my head. So that is a really good training, how we can convey plant breeding and genetics and the approaches that we're using to the masses that are outside of STEM. So I think those small talks with your Uber drivers, with your waiters, even with your bartenders could go a long way with that kind of training. And if you want to go for a more structured situation like that, there's always Toastmasters. So yeah.
You participated in Toastmasters?
Not really, actually, although my roommate before has participated in that, and we usually share some
Mhm.
insights about, public speaking. So yeah.
That one's been on my bucket list to try. I just haven't quite gotten the time yet.
Yeah, I would also personally want to try that at least a couple of times.
So we've been talking a lot about staying current. Do you have any suggestions for improving technology adoption across breeding programs? Sometimes it's very easy to get siloed into your own breeding program and focus on delivering the pipeline. And it's difficult to find the time to do the research to find new technologies and do the proof of concept work to bring them in. Do you have any thoughts in this space?
Quite honestly, technology adoption is always one of the bottlenecks in the scientific community, not just in plant breeding. So the most important thing, like you mentioned, is really finding that time for you to actually test out the system that you're developing, the approach that you're developing, really have that beyond just proof of concept. Because I think in any adoption, regardless if it's in plant breeding, regardless if it's engineering or whatever, the most important thing that the other scientists will look at, your stakeholders will look at, in our case, the farmers, the extension specialists, the agronomists will look at, is, you know, data, proof. evidence that our approach is working. If it's a prediction approach, okay, we should go beyond just cross-validation. We need to see it applied in a selection sense. If it's variety we're trying to promote, then if I'm a farmer, I would want to see the performance of this compared to the other existing varieties. And what does your data say if I plant this in this kind of environment, in this kind of environment, in this kind of disease pressure? So the most important thing for me for technology adoption, regardless both on the basic, translational, or applied science, is evidence. I think that goes a long way.
And what do you do when you've invested a bunch of time and the evidence says, this is not the approach to pursue? What do you do? How do you make that pivot? Any recommendations there from your experience?
It's really hard to let go, especially if you spent your time and energy working on something. But first, we have to accept that, not everything will have positive results. But I like to think of it as a learning experience as well. Because, okay, now I know that this approach is not working. Now I know that this product is not working. So I'm not going to make the same mistake. Now I can look at, okay, maybe I could try this approach instead. Maybe I could develop the product this way. Maybe this product will be better than the other one. So those failed experiments, if you will, actually for me is not failure. Those failed products are not failure for me. Those are just learning opportunities that, if you use it correctly, could actually propel you to better things and, better approaches and better results.
I like that outlook. Well, my last question for you is, if you weren't in plant breeding, where do you think you would be? How would you be applying this love for translational applied work and communication?
Oh, yeah. This is actually one thing I haven't thought deeply, but just to go back again, story time. Uh, back in the Philippines, when you apply for a university, you do entrance exam, and in there you always have at least two or three courses of choice, if you will. So my first choice is always, of course, biology. My second choice is chemistry. So if not biology, maybe I'm a chemist, or if not plant breeding, sorry, I may be a chemist. But the third one is veterinary medicine, which is really interesting. Um, but somebody told me, which is why I did not pursue veterinary medicine, other than I realized I really am bad in animal physiology and anatomy. Somebody told me I have to tip a cow to pass that course or to pass the degree. And I'm like, look, I'm gonna be trampled by a cow, so no, maybe veterinary science is not for me. Apparently, he was just joking, but I took it really seriously. But outside of STEM, I feel like I could probably be in something related to language and literature. Because when I was in high school and even in my elementary days, I was always participating in essay writing contests, in, speech contests and stuff. So I feel like if STEM did not work out for me, I would be in something related to language and literature. I don't know if it's writing, I don't know if it's speaking, I don't know if it's producing something, I don't know, but I'm pretty sure it's along that field and along that lines.
Well, I can see there's definitely a potential in the future for those to converge, for the
Oh
plant
yeah,
breeding and literature and outreach to converge.
that is true. Yeah, that is true.
I'm really enjoying the mental image of the cow tipping, steering you away from veterinary medicine.
I know.
Well, thank you so much for taking the time to share your work with us today. This was really wonderful to get to chat.
Well, of course, again, thank you for having me and thank you for this opportunity. I really appreciate it. And I really like sharing, my perspectives in plant breeding. I really like sharing what I'm doing. So, yeah.
Thank you so much.